These lesions may be caused by infection with low risk as well as high risk HPV, which undergo the processes previously described in the life cycle of HPV. Low-grade lesions are characterized by mild morphologic changes to the cellular structure and organization of the epithelium. The grading of the lesion is based on the level of epithelial abnormalities. For example, in CIN1, abnormal cells are concentrated in the basal third of the cervical epithelium. In addition, enlarged keratinocytes known as koilocytes can be found in the upper layers of the stratum spinosum and stratum granulosum. These cells serve as markers of HPV infection and are characterized by a cave-like vacuole surrounding an enlarged nucleus and an irregular nuclear envelope. The morphologic features of CIN1 can be detected as mild abnormalities on Pap testing. Approximately 35 to 40% of CIN1 is caused by HPV types 6, 11, 16, and 18. Other benign lesions known as condylomas or warts may occur on the vulva and perianal region or on the mucosa of the vagina, urethra, or cervix. Expression of the HPV proteins, specifically E6 and E7, by the infected host cell increases the frequency of basal cell replication and disruption of the normal maturation of the squamous epithelium. As a result, all epidermal layers above the basal cells proliferate, leading to a thickening of the stratum spinosum, stratum granulosum, and stratum corneum, also known as hyperplasia. In addition to the thickening of the epithelium, an upwelling of the basement membrane occurs because of the proliferation of connective tissue and the expansion of the capillary system in the dermis. These events are necessary to support the proliferation of the epithelium. The visible abnormal growths composed of a thickened epithelium are recognized as genital warts, which can be found on the external genitalia or mucosa of the cervix or anus. Genital warts are most often associated with non-oncogenic HPV types 6 and 11. High-grade HPV lesions. In contrast to low-grade epithelial lesions, high-grade lesions are caused by high-risk or oncogenic types of HPV, which can lead to the development of precancers or cancers of the cervix, CIN23, vagina, VIN23, and vulva, VIN23. The development of high-grade lesions is also characterized by a different sequence of steps in the life cycle of HPV from that described previously. Most often, infection with high-risk HPV types is associated with integration of viral DNA into host DNA. The early viral protein E2, responsible for inhibition of the genes encoding for the E6 and E7 proteins, becomes inactive, leading to overexpression of these proteins. HPV-infected cells that overexpress E6 and E7 proteins extend their lifespans and retain the capacity to proliferate, a process known as immortalization. Normal cellular mechanisms that protect the cells from mutations are disabled. As mutations accumulate, the cells become increasingly abnormal and undergo morphologic changes that lead to advanced stages of neoplasia recognized as CIN23, a cancer precursor and carcinoma in situ, and in some cases, invasive cancer. CIN23 and invasive cancer are most often associated with oncogenic HPV types 16 and 18. Unlike low-grade lesions, there is little to no expression of the late viral proteins L1 and L2, and there is minimal to no viral assembly and release of viral particles. Approximately 630 million people are infected with HPV worldwide. The annual incidence of low-grade dysplasias is estimated to be 30 million cases worldwide. Incidence and prevalence data for genital warts are more difficult to ascertain. Approximately 10 million cases of high-grade dysplasias occur globally each year. Worldwide, cervical cancer is the second most common cancer affecting women. Approximately 80% of cervical cancer cases occur in developing countries, and cervical cancer is the second leading cause of cancer death in women. Globally, approximately 500,000 women are diagnosed each year with cervical cancer, and almost 300,000 die from the disease. Despite the tremendous impact of screening on cervical cancer morbidity and mortality in developed countries, cancer remains an important medical issue worldwide, and warts and benign cervical lesions a significant medical burden.